Begin transmission. I'm supposed to talk about crustaceans today, but I always have a hard time talking about crustaceans because they're my least favorite arthropod, and at this point I'm always really excited to talk about insects because there's so many insects. And so how can I justify talking about lobsters when there are assassin bugs to talk about, right? But I have to talk about crustaceans a little bit. We already have in lab, and there are some fascinating crustaceans to be sure. The gooseneck barnacle story is fantastic, and the peacock mantis shrimp is one of my favorite animals. But when I think about crustaceans, all I can think about is let's get to the insects. So eventually we will, but we got to get through crustaceans first, and then chelicerates, and then we can talk about some fascinating insects. So to spend some time on crustaceans, we'll do that today. And I want to mention just one particular crustacean that I find especially fascinating, and that is, you guessed it, a parasite. There are plenty of parasitic crustaceans, and one of the most disturbing stories can be found in a genus called Sacculina. You can see it here. The crab is a normal-looking crab, but on the underneath, on the ventral side, you see this kind of bulbous sac. That right there is a parasite. And it is a relative of barnacles, and so it is a really good example of the transition from being free-living commensal organism to being parasitic, so kind of this deterioration of relationship, which I find incredibly interesting. But these Sacculina parasites, what they do is as larvae, they find a crab host and they wander around on the crab until they find a joint, and then they burrow into the joint and as they molt and then transform into an adult. They migrate to the anterior and then kind of exude themselves into the outside world. And then in this picture here, this drawing, you can see that the parasite actually has two different parts to it. It has an externa, which is this bulbous sac that lies outside the organism, and then it has an interna, which are these kind of dendritic thread-like things that are internal of the host. And when you look at this creature, it took us a while to figure out this was actually even an animal, that it was a living thing in its own, because it doesn't have legs and it doesn't have eyes, doesn't have really much of a nervous system even. It is just this completely degenerate little blob that takes complete control of the behavior and resources of its host. And the only way we know it is a barnacle is because the larvae are free-living and the larvae look very barnacle-y. So we know it's kind of a derived barnacle that's kind of adapted specifically to parasitism. But here's the crazy thing. The interna, these dendritic threads, those kind of form roots, and they just envelop all the internal organs of the crab. So some go to the liver and the stomach and just seep nutrients from the crab. Others integrate into the brain and nervous system, and they take mental control almost over the host. They control the reproductive system and the hormonal cycle of the crabs and even their behavior. So what happens is if it gets into a male crab, well, before I get to say that, so crabs and crustaceans, as you know, they usually have some type of brooding behavior for their offspring, so they have some maternal care, and the mother will make sure the eggs are washed with oxygen and has plenty of nutrients. Well, the crabs do this too. They usually have a little brood sac on their bellies, and the little baby crabs they keep fresh and taken care of on their ventral side. So it's no coincidence that the externa of Sacculina is on the underside, the ventral side of the abdomen. That's exactly where the crab's offspring would normally be. And so when the parasite infects a female crab, it activates the brooding behavior of the female. And so the female takes care of it and nurtures it and makes sure that when the Sacculina larvae are born, they're kind of pulsed out into the water, and then they're taken care of. So it transforms the behavior of the crab, initiates brooding behavior. It also eliminates molting because, as you can imagine, if the crab molts, it's gonna shed the parasite. So the parasite prohibits molting, activates brooding behavior. So what is it going to do with males? Because males don't brood. Well, simply it just transforms the males into females and then activates their brooding behavior. So it squeezes the testes out with these little tendrils and changes their hormonal system. So the males basically take on attributes of females morphologically and internally, and then they activate the brooding behavior. So this is what's known as a parasitic castrator. It takes the females, makes them very maternal, and takes the males and makes them females, and then makes those new females very maternal. So that's a curious story. Just wanted to relate that one story to you. And now let's learn about crustaceans. Good morning. We're here to talk about crustaceans. Crustaceans are unique among the arthropods in that they are primarily aquatic. So the vast majority of arthropods are terrestrial, but crustaceans have maintained a tight connection to the water. So we have some familiar crustaceans like lobsters and crabs and shrimp, as well as some unusual small microscopic ones and some very strange parasitic ones that don't look like crustaceans at all. But to get started, what are crustaceans? So we know these are arthropods, and I just said that they're primarily marine. There are some freshwater species, but the vast majority of species are marine, with a few terrestrial ones. This one here is a cleaner shrimp, cleaning the leftover food and bacteria and parasites from the mouth of a moray eel. And so a lot of times crustaceans form tight relationships within their community, whether it's cleaner shrimp or crabs and the sponges that they put on their backs, or even that earlier in the semester we talked about the pom-pom crab that uses the sea anemones in his little claws as protective little stinging pom-poms. So crustaceans are important members of marine ecosystems, particularly coral reefs, where they are primarily predators, although there are a few herbivorous species. And on land, you have things like rolypolies, armadillidium species that are primarily detritivores. So all kinds of different lifestyles, from predators to herbivores to parasitic. We have some really tiny ones, so these are little cyclops, and they're also really important members of zooplankton, which are kind of suspended tiny creatures in the water column that form the very basic component of marine food chains. So the zooplankton feed on phytoplankton, so I guess the zooplankton aren't the basic ones, but phytoplankton and the zooplankton, and then all basically everything in the ocean relies on zooplankton for sustenance in some way or another. So when we talk about all the animals that filter feed in marine environments, they're primarily filter feeding zooplankton, and a large component of zooplankton are microscopic crustaceans, like these guys and many, many, many others. There's also some that are a little disturbing looking. Crustaceans are the crunchiest of the arthropods, and this particular one is a marine rolypoly. So it's an isopod related to our friendly little rolypolies, and it looks quite intimidating. These guys can get over 2 feet long and with a pretty substantial shell, and we've seen them in swarms devouring whale carcasses. So they can be quite voracious scavengers. So as a little palate cleanser, here's a cute little adorable baby crab, and this is a red crab, and he lives on a place called Christmas Island. And every year there's a famous migration of crabs on Christmas Island. All those little red fuzzy things all over the rocks, those are millions of tiny crabs that migrate from one end of the island to the ocean to live. So some pretty remarkable. If you're ever in the area, make sure to visit Christmas Island. Crustaceans can be distinguished from Chelacerata and Hexapoda by several different traits, and really all of these subphyla have really distinct characteristics. So I'll expect you to be able to identify a new creature based on just a list of synapomorphies, right? So here we have a crab and a mantis shrimp, both crustaceans, and to distinguish crustaceans from Chelacerata, we already know that they have mandibles instead of chelicerae, And so that kind of makes them seem like hexapods. So mandibles belong to both Hexapoda and Crustacea. However, crustaceans only have, or I should say hexapods only have a single pair of antennae, while crustaceans have two pairs. So they have two pairs, one for kind of equilibrium and balance, and the other one for your typical antenna qualities of sensing chemosensory things in the water, smelling, tasting, touching, that kind of thing. They also have a really elongated carapace that covers most of their body segments. So their carapace kind of extends from their thoracic cuticle and extends posteriorly across their lower segments. And so you get a hardened carapace. In some species, this is reinforced with calcium. So particularly in crabs, they have a really hard calcium reinforced shell. So it's much tougher and thicker than the shells of hexapods, and especially chelicerates. Have you ever poked a spider butt? They're kind of soft and squishy, FYI. Inside their mouth, they have all kinds of different mouth parts, maxilla and maxillipeds, and three maxillipeds and 2 maxilla, and also a mandible. So in their head, we can see this tagmata that's been fused. These segments have been fused during development, and now they have lots of specialized appendages for filtering food and water. Probably the best synapomorphies are going to be the double pair of antennae, and then also the biramous appendages. So if you're looking for the two best synapomorphies, it's going to be the double pair of antennae and the biramous appendages. And just as a reminder, the biramous appendages mean that is referring to the branching of their appendages, and it's in contrast to uniramous appendages that belong in Hexapoda and Chelicerata. And Myriapoda. I always forget about the myriapods, but they're important creatures too. Crayfish appendages, this one should be a little bit of review. At the top we've got, this is a crayfish, but this can apply to lobsters and shrimp as well. They all have very similar morphologies, just kind of larger or smaller. Their first pair of walking legs is called their chelipeds, and they have an elongated chela or claw that they use for defense and offense, for grabbing prey, for manipulating prey into their mouth. And then moving posteriorly, we see all their little mouth parts that I was talking about. And this is probably the best example of fusion of segments and then the differentiation of the appendages that used to be on those segments. So we have all these different maxillipeds and maxilla that are adapted for crunching food or mashing food or filtering food or tasting food, or bringing in fresh oxygen and water to their brachial chambers. So they all have really specialized mouth parts that are all kind of smashed up together. And then they have 4 more pairs of walking legs, so 10 in total. Crayfish are in the Decapoda, along with the crabs and their relatives. So 10 pairs of walking legs, and that's in contrast to chelicerates, which usually have 8, and hexapods, which usually have 6. And as I mentioned during lab, you can distinguish the second and third walking legs from the fourth and fifth by their little claws at the tip. So the second and third walking legs still have little tiny chela at the end, little tiny pincers that they can use to help manipulate food. And then you have swimmerets. And also, as you know, the very first pair is the external morphology that distinguishes males from females. The males will use the copulatory swimmeret to transfer sperm to the female, and then the female uses her swimmerets as brood chambers, so the eggs will be adhered to the little swimmerets, and she'll keep them nourished and oxygenated and fresh until they hatch. And then at the very end, we have the uropod, which is kind of a nice long flap that can be, when the abdomen is curled under and retracted, it can lead to a quite rapid backward propulsive movement as the crayfish swims backwards. Ecdysis is the process of shedding one exoskeleton for a new one growing underneath. And we know about this a little bit. We know that these are enclade ecdysoa, and we know that this is hormonally controlled by a hormone called ecdysone. And we also know that the exoskeleton is expensive to make, and it weighs a lot. So we talked about that last time with characteristics of arthropods. It's expensive, and it kind of restricts their growth, their size, because of how much it weighs in proportion to their body. And so if you have something that's really expensive, you don't want to just make it and then throw it away, because you're essentially just tossing aside all that energy, metabolic energy and physical resources, the calcium and all the ions and inorganic salts that you've put into the cuticle. And so what happens is pretty remarkable. As the crayfish grows, or as any of the arthropod grows, there are little specialized neurons throughout their whole body called stretch receptors, and they can actually tell how much the cuticle is stretching under the underlying growing body of the crayfish. And when these stretch receptors, when enough of them are activated, then it starts a cascade signal pathway through the X organ and Y organ. So to understand the process of ecdysis, you need to understand it starts with stretch receptors being stimulated, being signaled that the underlying body is stretching the cuticle to an uncomfortable level. And then the X organ is what produces molt inhibiting hormone, and then the Y organ produces molting hormone, or ecdysone. And so you kind of have to think about it in this way. The Y organ is kind of always wanting to molt, and the X organ is just this kind of barrier, this dam that prevents the molting hormone from flooding the body of the crayfish. And so the X organ's job is to prevent molting, and so it's always on until the stretch receptors send signals to it that say it's molting time, and then the X organ shuts down, stops producing molt inhibiting hormone, and when molt inhibiting hormone, the levels decrease, then that allows the Y organ to start producing molting hormone, or ecdysone. So stretch receptors get stretched, that signal the X organ to stop producing molt inhibiting hormone, and when the threshold of that hormone reaches a certain low level in the body, the Y organ is then freed to flood the body with molting hormone, or ecdysone. And if you look over here on the right, we can see that we have an underlying epidermal living tissue, and just like we've seen from everything from sponges to here, you're going to have living tissue that secretes a non-living tissue. And so this epidermis secretes the cuticle, and if you look really closely at the cuticle, you're going to see two different parts to it: an epicuticle on top, and then a procuticle underneath. And then if you look even closer, you can subdivide the procuticle into an exocuticle and an endocuticle. So the epidermis secretes everything. The epicuticle is on the very top, and then the procuticle has two components: an exocuticle and an endocuticle. What happens during molting is that all the old cuticle separates from the epidermis, and the epidermis starts secreting a new epicuticle. But if here's the remarkable thing: all that old cuticle is expensive, it's valuable, and so what the crustacean, what the arthropod does, is it dissolves the old procuticle, the old endocuticle part of the procuticle, and then reabsorbs all those inorganic salts and proteins as much as it can. And then it uses those, it recycles those into the new cuticle. So eventually the things that can't be reabsorbed, the old epicuticle and exocuticle, that gets discarded, and then you have this nice flexible, fresh cuticle that needs to be stretched and hardened, and that's the process of ecdysis. Here you can see a crayfish wiggling out of its old epicuticle. Pretty remarkable, and it's so clean and fresh and beautiful. Wouldn't that be nice if every few months we could just completely shed our old selves and put on a new self that's clean and perfect and whole? That would be nice. Very interesting process. And so with crawdads, it splits down the back, and then they kind of wiggle out the back. And it's just remarkable to me that they can get their antenna through there and all their little legs, and it's just most of the time it works out very well. So at this point, it's really soft and squishy, I've actually accidentally caught some crawdads when they were a little squishy and just mashed them all up. So usually at this point they'll find a nice, dark, safe place to hide for a while until the process of tanning and sclerotization are complete. So that's the process of the hardening of the epicuticle. Here is one of a crab doing this, which is kind of even more remarkable to me because it's got these 8 very, very thin legs, and this is sped up quite a bit, but it can pull all of those out of their old little exoskeleton. And just also for your enjoyment, here's a little spider doing that, because spiders also shed their exoskeletons. And often you can find these leftover skins on the forest floor or even on tree branches. They're pretty common. Pretty fascinating process. All right, the nervous system and the endocrine system. We kind of have been talking about the endocrine system already. This is the system that controls hormones. So molting is endocrine system controlled. And the endocrine system is also, it's tightly tied to the nervous system. So in the Y organ and the X organ, you have specialized cells called neurosecretory cells, and these are going to be what secretes the hormones. Neurosecretory cells elsewhere are going to control the rate and amplitude of heart rate, and they're also going to control the amount of pigments in the chromatophores throughout the body. So the endocrine system is responsible for molting, for heart rate and amplitude, and for pigment changes. And you also have some specialized glands called androgenic glands, and these glands are found in male crustaceans, or I should say male crawdads at least. I don't know about the rest, but these androgenic glands are responsible for the formation of testes and then male sexual characteristics, so the copulatory swimmerette primarily. And the fascinating thing here is if you take an androgenic gland from a male and put it into a female crustacean, then at the next molt, she will take on characteristics of a male, because those male characteristics are tied to the production of the hormones from the androgenic gland. So she will begin to, her ovaries will transform into testes, and her swimmerettes will transform into copulatory swimmerettes. So very interesting, there's a tight correlation between the androgenic gland and male characteristics. The nervous system itself is highly controlled by a supraesophageal ganglion, known as the brain, so that's the largest clump of neurons. But you can also, as you look down the ventral side of the crayfish, you see clumps of ganglia. You have a subesophageal ganglion that's going to be below the esophagus, and that is going to so what you notice is that those ganglia scattered throughout. Those are going to be where the legs intersect with the body. So at the junction of every segment where you have paired legs coming off, you're going to have a clump of neurons to interpret all the senses that you're getting from your legs. So a lot of the legs have taste receptors, and a lot of them have chemoreceptors or mechanical receptors, obviously, and all that signal gets interpreted and then sent to the brain with these little clusters of ganglia. Also, you may notice if you look at the esophagus itself, there's a little circlet of nerves around that. We saw that in mollusks as well. So the crustacea digestive system is also encircled by nerves. I should also mention something called giant axons. These are thicker, longer neurons that can conduct information really, really rapidly, and so they're the fastest transmitting neurons in the body. Giant axons. Sight in crustaceans is really important. For most arthropods, they rely on visual information quite a bit, as well as chemosensory information. But in crustaceans, you can have compound eyes and nauplian eyes or median eyes. And we've talked about median eyes a little bit before. These are what's found in cyclops and a lot of smaller crustaceans. They essentially are composed of something called pigment cups, which have retinal cells in them. So it's fairly simple. kind of analogous to ocelli. They're good at detecting motion and light and dark, but not really. They don't form images, and they don't give depth perception. In contrast to this, compound eyes are composed of ommatidia, and compound eyes can either be sessile, like you see on this little rolypoly relative, and sessile compound eyes means they're attached directly to the head, or they can be stalked, and so you have little eye stalks like on this crab, and you can see the compound eyes are on stalks. What's the benefit of being on stalks? It gives you a wider field of vision, and you can rotate them so you can see a little bit better. What's the benefit of being sessile? Well, your eyes aren't sticking out of your head, and so you don't get chopped off or something. They're more protected. So compound eyes are composed of ommatidia, and the ommatidia have thousands of little facets in there, and these ommatidia are they have a lens that's going to direct the light into these long columns that are surrounded by pigment cells, kind of columnar pigment cells. And so it's a very different kind of eye than the image-forming eye of mollusks and vertebrates. But what it's good for is wide field of vision and detecting motion. So they're really good at detecting motion, and also they can see polarized light. So the compound eyes with these ommatidia, sometimes 15,000 or so ommatidia in a single compound eye. They are good for detecting light, wide field of vision, and detecting polarized light, which are all three things that they're better at than vertebrate or squid eyes. Crustaceans breathe with gills. They are underwater, so they're not going to have tracheal systems like the hexapods, and hexapods breathe with a really advanced tracheal system. Little known fact, spiders get most of their energy through books. We'll talk about that later, but yeah, it turns out reading is a pretty good way to get energy. Not really reading, but they do get energy through books. Crustaceans breathe with gills in brachial chambers. So on either side, on both lateral sides, they're going to have a special protective section underneath their carapace that is a little chamber that houses their gills. Water flows across them, and oxygen gases are exchanged at that point. They're going to be highly vascularized. They're going to be they're going to have maximum surface area, and you can see here on the right that they are kind of found at the base of every leg. Waste is secreted through specialized glands called antennal glands. Sometimes this antennal gland is called the antennal gland because it's at the base of the antenna. Sometimes you'll see it referred to as the maxillary gland because it's also right at the maxilla. Sometimes it's called the green gland because it looks a little greenish. But regardless of what it's called, this antennal gland is used for secreting metabolic wastes. So this is going to end ions. So this is their kind of osmoregulatory excretory system. And they have a complete digestive system and an anus in their telson. So solid waste is excreted there. But then liquid waste, ammonia and other ions that they need to get rid of, that's going to be secreted right at the base of their antennae. Now, how does this not end up with fouling? Because you have waste secretion both at the head and the tail. So this is kind of interesting. So for solid waste, the crustacean can just walk away from it. So there's no fouling there. But what about the liquid waste here from the antennal glands? You're excreting it right into your mouth, which seems the opposite of what you would like to do. Well, how do crustaceans move rapidly? They move backwards. So they can just retract their tail and then propulse backwards. And so if they're secreting from the front, they can propulse backwards. If they're secreting from the back, they can walk forwards. So they're able to avoid fouling either way. The crustacean circulatory system is an open system, and we've talked about the heart being controlled by the neurosecretory cells. and this is going to pump blood, and then it's just going to bathe the hemocoel with blood, and they actually have respiratory pigments that are going to carry the blood to specific areas. So the respiratory pigments are usually called hemocyanin or hemoglobin, and you can see here they're really interesting structures and just perfect for carrying iron and blood. Oxygen, iron and oxygen. So the hemocyanin looks blue, cyanin. The hemoglobin looks a little bit red, and hemoglobin is also found in vertebrate respiratory systems. But the hemocyanin is pretty unique to crustaceans and their relatives. The most highly vascularized part of the crustacean is the gills, the brachial chambers. That's where the respiration is going to happen. So that makes sense. I mentioned earlier that crustaceans are some are predators, some are herbivores, some are parasites. There's also some that are filter feeders. So sessile ones like barnacles are primarily filter feeders. They have specialized cirri, specialized little legs with long bristles that they use to filter the water for food. Others are active hunters, and they'll actively seek out their prey. And usually if you're a predator, you're going to have something in your stomach called a gastric mill, which are basically calcified teeth that they use to help mechanically break up their food. This is a crab who's caught a poor little baby sea turtle. Very sad. And then here is a mantis shrimp. We talked about mantis shrimp like Squilla that have these giant thumping claws that they use to break open shells of their prey. There are other species that have these spear-like cheliceds that they use they have these little burrows, and then they lash out and spear their prey really rapidly. Pretty remarkable. Faster than we can even see, that's why this has to be slowed down 60 times. There also can be quite neat eaters. Look at this adorable little crab, so delicately. With, you got these massive cheliceds, but he's so graceful. Type of guy you could take home to your grandma, and they love spaghetti. Reproduction in crustaceans is usually going to occur in the water, and they're almost all dioecious, except the barnacles, but we'll talk about that in a minute. They usually have a protrusible penis that they use to transfer sperm from the male to the female, and in crustaceans, in crawdads, crayfish, and decapods like that, they're going to have copulatory swimmerets that perform that same function. And then here's a giant mating swarm of crabs. So at the bottom of the ocean, sometimes they'll get into giant mating swarms of male and female crabs, and they'll all mate together and then lay eggs in giant broods. Once they lay eggs, there's usually some kind of parental care. Usually the mothers will have a brood sac or something. These are little tiny crustaceans called water fleas, and you can see in the back here the little eggs that are being cared for by the mother. We know that terrestrial rolypolies, armadillidium species, have marsupiums, and we know that crustaceans have little brood sacs on their swimmerets as well. One thing I'm just noticing here, what is this thing in the picture? We got crabs mating, and then we have rocks, but this looks unnatural, right? What is that? I think that is actually made from an insect called a caddisfly larva makes that, and the caddisfly grabs a bunch of pebbles and stones that it likes and then glues them together with a silk, and then it makes kind of a hollow cylindrical tube, and it lives inside that. But that's not really important. That's just an interesting thing I noticed. Pretty cool construction, though. So what about the monoecious species? So barnacles are monoecious, and that presents, so they have male and female parts, and then but they don't want to self-fertilize, so usually they practice cross-fertilization. We've seen this a lot with platyhelminthes that are also hermaphroditic. But there's a particular challenge to cross-fertilization for barnacles because they're sessile. So how do you mate with something if you don't move? So you're familiar with some organisms that are sessile and do something called broadcast spawning. So think about sponges and sea anemones and even some annelid worms. The broadcast spawning is when they release the sperm and the eggs into the water and hope for the best. So that's one solution to mating if you're monoecious and also sessile. But crustaceans usually have some kind of sperm transfer organ, and so they have a protrusible penis. And so if you already have that, you don't want to necessarily do broadcast spawning because you already have the resources for more intentional sperm placement. And so if you think about pros and cons of that, broadcast spawning, you have to make an enormous amount of gametocytes for low chance of success. But if you have a penis, you don't have to make as much sperm, but you have to have a penis. And then you have to find a mate. So how do barnacles find a solution to this difficulty? Well, some of them go one route, and they don't really develop any penises, and they do something called sperm casting. So it's very similar to broadcast spawning, except it's only the sperm. So the sperm gets released, and then presumably the series of females are able to, or they're monoecious, so the series of other barnacles can capture the sperm from the water. The other way around this is to double down on the penises, and instead of sperm casting, just have an extremely long prehensile penis. So some barnacles, some barnacle species have the longest penis to body length ratio in the animal kingdom. Sometimes their penises are 8 times as long as their bodies, and they're prehensile. They have tactile receptors on them and little bristles, and they can just kind of feel their way around, because again, they're sessile, and so the penises kind of extend out and then feel all around until they find another barnacle, and then they can mate. So kind of strange, but makes sense as a solution. So you can either do sperm casting, or you can just have an extremely long prehensile penis. That's how barnacles mate. We like to eat a lot of crustaceans. Crabs and lobsters and shrimp are pretty famous delicacies. And one of the most common ones that we eat is the Gulf Coast shrimp, seen presumably here, although I am not 100% sure actually. But next time you eat shrimp, think about what you're actually eating. So at the back here, the crunchy part that we don't normally eat, right? That's the telson and the uropod, and then this is the abdomen. And so if you see a dark line running along the dorsal side of that, that's going to be it could be the dorsal blood vessel, or it could be the intestine. The intestine is going to be filled with shrimp poop. So if you eat shrimp, you're getting their digestive system and everything, unless that's been peeled out, but it often is not. At any rate, their mussels taste pretty delicious to us, and we eat millions upon millions of these every year in the United States alone. The Gulf Coast shrimp is found in the Gulf Coast, not surprisingly, and it starts off as adults, which lay eggs, and then you have 3 different larval stages before they molt into their juvenile stage, which looks a lot like their adults. You have a nauplius larva, and then a protozoea larva, and then a mysis larva. And look at what they look like. They look like nothing. They look like something, but they don't look like shrimp. So think about metamorphosis. Here's a picture of a nauplian larva, and this is where the word nauplian eye comes from, because the nauplius larva has a little median eye on it. But you can imagine that it's taken a long time for us to associate these larvae with their adult forms, and so there's still probably a whole lot of animals out there that we have misidentified. We don't know which larvae which go with which adults. And I want to also point out that the protozoa larva, why is it called that? It's because it looks like some weird kind of protist, some kind of weird unicellular organism. So a lot of these historically have been considered to be their own species, and it's been within the past 50 years or so that we've been able to identify these complete life cycles, primarily when we start reproducing them we culture them for food. So the ocean is full of some really strange creatures for sure, but know this general life cycle. It's pretty easy. You have adults that lay eggs, 3 larval stages: nauplius, protozoa, mysis, and then you have a post-larval stage and a juvenile stage. These are all characteristic. They just molt into the next one, and so they're not parasitic on anything, and so pretty easy. In lab, we've kind of talked about this, but here's just a general overview of the classes and the orders that we'll talk about. Remember, this is Subphylum Crustacea, so this is going to include 5 distinct classes. The pentostomids, which include some parasitic creatures, the brachiopods, which are going to be your small triops type of creatures, your copepods, which are also really small. Some of them are important intermediate hosts of parasites. Then you have barnacles and thecostraca, and then the malacostraca have your isopods, amphipods, and our most famous decapods. So most of the familiar crustaceans you know and love so much are in class Malacostraca. And these are crustaceans. Brief update on our mysterious death investigation. So let's just, I just want to take through our suspects one at a time. And so I've kind of eliminated from contention at this point myself, Charlie, and Master Sergeant Rush, although we can at any time go back and reinvestigate any of those people, which leaves 6 other people as potential suspects. So I'd just like to kind of think out loud about each one and see if they have any potential motivation and see if they have any cause for further suspicion on my part. So I'll begin with Isabella, because Isabella is kind of the closest to Charlie and me. She hangs out with us a lot, and particularly Charlie, because apparently they got some type of romantic interest. I guess we can't really have a story without some romantic tension. It's a good drama builder, I suppose. But Isabella is she's the daughter of the captain, which is also kind of odd, right? This is a 10-member crew, and we have Charlie as my son, and then Isabella is the captain's daughter. So it's a very tight, close-knit community here. And I guess the only thing I can say about that is, I didn't put this crew together, right? So this is all put together by the International Space Exploration Agency. The ISAE, as you know, because that's you guys. So they're not here because they are our offspring. There's no nepotism here, at least not on my part. Charlie and Isabella are adults. They're both around 19, let's say, and fully capable of being members of this crew on their own. They just happen to be related to 2 of the other members there. And it kind of makes sense, this is the 19th mission. We're really not the best of the best anymore. We're just kind of the best of the rest, if you will. After the 15th voyage, when all the crew members got disincorporated as they passed through the vortex of the nebulari, you remember that, as is quite traumatic for everyone. We didn't think there would ever be any other voyages, but they found enough volunteers for a 16th, 17th, 18th, and now 19th mission. So all that to say, it's fine. It makes sense why we have two children of two crew members on the 10-member crew. So it all works out. So anyway, Isabella's role here is she is kind of our survivalist, problem solver. She's studied extensively about different worlds and Earth's history, and she knows about using plants and animals and resources, locating them, and she's just a general survivalist, which is good to have when you're going to a new planet. So that's one point of suspicion, is that she's good with problem solving. So she would be capable of, and she's good with natural history. So she would be capable of discovering some natural product in nature and then using that toxin or venom or protein or whatever to achieve a certain end. So Isabella has some skills, some murdery skills, I guess. She has some skills that can transfer pretty easily into some nefarious purposes. Although her character, I think, is pretty high. I wouldn't approve of her relationship with Charlie if I didn't think she had a high character. Although I don't approve of their relationship for some reasons. It's very complicated, and the captain is quite opposed to their being together. and that leads to some tension. And I think a while ago there's a bit of a squabble about that, and the captain forbade, as it goes, Isabella and Charlie from seeing each other. Now, I talked to Charlie about this, and Charlie said that because he's honest with me, he's forthright, he said that Isabella and him have been seeing each other after the captain told them not to, secret trysts and all that. But the captain's wise, and he monitors communications. He's the captain. So how were they meeting together? How were they communicating in secret? Well, Charlie said that they were actually communicating through Dr. Mastronomer. Mastronomer was a compassionate soul on their predicament, and agreed to kind of ferry information and help them kind of meet in secret away from the captain. I, of course, didn't know anything about this. I'm pure in this regard. So apparently Isabella and Charlie were seeing each other after the captain told them not to, even though they're adults. They're 19, but captain's got his reasons, and they were using Mastronomer as a go-between. So there's something there potentially. Isabella has the means to commit murder, but I am suspicious of her character to do so, and we do have a situation where Mastronomer was put in a situation that was a little tricky. So I know there's quite a bit of tension between Isabella and her father about this. Perhaps if I could speculate wildly, which I can do because this is just to me and you, just Josh and Alfredo hanging out talking about our theories about murder, and so perhaps what happened is that Mastronomer was convinced that she needed to stop helping them, and went and threatened to go to the captain, and then Isabella nixed that. So that's my suspicions of Isabella. She's got means. I could at least imagine a motive. And so Isabella is not removed from consideration of suspicion at this point. Next on our list is the captain, and we'll talk about the captain next time in transmission.